All right, I'm going to take up the homework on the lac operon and gene regulation on page 339. The question number 1 says, define the following terms, and so you have an operon. So an operon is it's a cluster of genes that contains a DNA sequence to regulate the production or metabolism of a substance. So I'm going to repeat that one more time for you. An operon is a cluster of genes that contains the DNA sequence to regulate the production or metabolism of a substance. Okay? An operator is different than an operon. So an operon includes the whole section of all of the things that control whether the gene is going to be on or off, read or not read. An operator is where the regulator binds. Okay? So we have a place on the operon where the repressor binds. Okay, so we've got the operator is the place where the repressor binds, and I showed you that on the lac operon. The co-repressor is needed to activate a repressor. Okay, so a co-repressor can be like a signal molecule. It binds to a regulatory protein, and it decreases the expression of operons and their genes. Okay. A repressor, you already know what a repressor is. It binds to the operator and it shuts down transcription of the gene. Housekeeping genes are genes that are on all the time because their products, their proteins are essential for the organism on a constant basis. All right, and an inducer is something that can cause the gene to be copied. So it's a molecule that initiates the production of enzymes, is my official definition there. A molecule that initiates the production of enzymes. Lactose is an inducer. Okay, so if lactose is present, it turns on the lac operon. Question 2 says, why do eukaryotes have a more complex system of gene regulation than prokaryotes? I would say it's because prokaryotes are less complex. We have so many more genes, so we need to make sure that they're not all on at the same time, because we would lose a lot of energy in that case. So we have more complex systems because we have greater demands, because we have a greater number of proteins that we're making. Question 3, what features do the lac and the trip operons have in common? So what do they have in common? Well, they both have operons. Okay, so they both have mechanisms to control whether lactase or tryptophan is going to be made or not made. How do the operons differ? They're exact opposites. So when lactose is present, the operon is on. When tryptophan is present, the operon is off. Okay, so hopefully you did some reading on tryptophan and the trip operon. When bacteria are exposed to lactose, that means they want to make lactase, and so they turn that gene on. When you and I have lots of tryptophan, we don't need to make any more. So this is why it's like exact opposite example. If you've got lots of tryptophan, you don't need to make more. So tryptophan acts as a repressor, and it shuts off, a co-repressor, and it shuts off the operon. All right? And then question 4 says, how does the lac operon regulate the production of the enzymes needed to metabolize lactose? So that is exactly what I went over with you yesterday in the note. So if you understand the lac operon, then you understand question 4. Question 5, describe the trip operon system if the level of tryptophan is low. And then it says include all the activity states. You don't need to worry about that. What I want you to worry about is just this first sentence and understand what happens if we change the amount of tryptophan. So if tryptophan is low, then the trip operon is going to be on, okay? Because there's going to be less tryptophan to bind to the repressor, and so the repressor won't be bound, and so we're going to have the copying of tryptophan, of the trip operon. So if trip is low, the operon is on. Why? Because you have a low abundance, you have not very much tryptophan, so you need to make more. If trip is high, it acts as a co-repressor. So trip then binds to the repressor binds to the operon, and then we can't make trip. Okay? So if you've got a lot of tryptophan, you shut the operon off. When there's less tryptophan, the operon turns on. All right, number 6 says, give an example of a eukaryotic regulatory mechanism that occurs during transcription. So during transcription, that would be a post-transcriptional modification. So I know they're saying during transcription. I would go with capping, tailing, and cutting out introns. During translation, we could say we're not going to allow met-tRNA to bind with the two ribosomal subunits. And after translation, we can prevent those structures from forming on the polypeptide. So we can prevent the polypeptide itself from, forming a secondary, tertiary, or quaternary structure. All right. Question 8. The twin mice in figure 5 on page 336 have one small but important difference in their agouti gene. What is the relationship between the phenotype of the agouti mice and the chemicals known as bisphenol A? So I like to give you this reading because it is really an application section of this genetic unit. These two guys should be identical, okay? This mouse here has an agouti gene. This mouse here has an agouti gene, okay? In most mice, the agouti gene is off, all right? And why is it off? Because it's methylated. So if you read this page, you would realize that methylation, it says right there, is another way to put genes or entire regions of chromosomes on hold. So if we don't want to be copying a gene, we methylate it for the time being. All right, and then when it gets to the right area in the cell or the right cell itself, then we would turn it on. So, because the methyl group would be taken off. So this mouse here has an agouti gene, all right, but it's not turned on. This mouse here has the agouti gene and it's turned on. So you can see the difference in phenotype immediately. The biggest problem with this mouse, it's a beautiful color, but it's obese, okay? And so when the agouti gene is turned on, this mouse becomes obese, and it becomes this nice ginger color, a nice orange color, okay? The problem with that is there are lots of risks, like with diabetes and cancer, when you start to get a mouse that is this obese. Now, bisphenol A is known to the public as BPA. So BPA is a substance that was in a lot of plastics, okay, for a very long time. In your lifetime, there was certainly BPA in your plastics. And then that's when people start to say, okay, I want a plastic that is BPA free, bisphenol A free. And so this is why you should check your plastic water bottles, okay, or just avoid plastic water bottles altogether, because BPA is known to turn on genes, especially in this mouse, okay? So not in every case, but in many cases, when a mouse is given BPA, the agouti gene is turned off, and then there are higher risks of cancer, obesity, and diabetes as a result of obesity, okay? And so they take that study and they say, oftentimes what happens is in mice happens to us, and so it's a warning. All right, so I thought I'd just show you that question and see that. And then the last question I gave you was on cancer. Discuss what happens to cells to cause them to become cancerous. So I already told you guys about telomeres and the fact that cancer cells have telomerase, so they're out of control when they're growing. Nothing regulates them. They don't ever go into cell senescence. Okay. The other thing about cancer cells is they're not regulated. Okay. So where I just taught you about the lac-operon and then eukaryotic gene regulation. So we have all those. We have 4 ways of regulating our genes, including methylation. Cancer cells lose those methods. So what happens is those methods become absent. The genes that are copied frequently are mutated in terms of the operons, and those guys are called oncogenes. So oncogenes. They're mutated and they don't control gene expression like the rest of our cells do. Okay, and so that's an additional problem with cancer is there's no regulation in growth. So in grade 10, you learned cancer grows just to grow, it just keeps growing all the time. And that's one of the reasons. It's because of those oncogenes. So your lac-operon, all those things would not be functioning. Okay.